Hey everybody, PreMedMama here. Thanks for joining me. I'm a mother and a pre-medical student. Today we're going to be in MCAT Biology Review by Kaplan. We're in Chapter 1 and we're looking at Section 1-3. I'm going to be talking about the prokaryotic cell structure, specifically the cell wall, flagella, and some other organelles. If you've been here with me on my videos before, thank you. If you're new, welcome. I'm so happy you're here. Please remember to subscribe and share this video and others if you want to. Thanks for your support. Let's get started. Prokaryotic cell structure. One of the main differences between prokaryotes and eukaryotes is that prokaryotes lack a nucleus and membrane-bound organelles. So they don't have a nucleus or membrane-bound organelles the way that eukaryotes do. Prokaryotes are also single-celled organisms, which means that each cell must be able to perform all of the functions necessary for life on its own. However, prokaryotes may live in colonies with other cells and may signal these cells to share information about the environment. How cool is that? We have an image here on page 17 of the MCAT Biology Review book by Kaplan, and it shows a diagram of a prokaryotic cell structure. We can see the cell wall, the cell membrane, a ribosome, the DNA nucleoid region, and a flagellum. Let's talk now about the cell wall of the prokaryote. Because prokaryotes do not form multicellular organisms, each bacterium is responsible for protecting itself from the environment. The cell wall forms the outermost barrier of the cell. The next layer in is the cell membrane or plasma membrane, which is composed of phospholipids, similar to that of the eukaryote. Together, the cell wall and the cell membrane are known as the envelope. The cell wall both provides structure and controls the movement of solutes into and out of the bacterium. This allows the cell to maintain concentration gradients relative to the environment. In bacteria, there are two main types of cell wall, a gram-positive cell wall and a gram-negative cell wall. The way that scientists can determine which type of cell wall a bacteria has is by using the Gram staining process. The Gram staining process utilizes the crystal violet stain, followed by a counter stain with a substance called safranin. If the envelope absorbs the crystal violet stain, it will appear deep purple color, and the cell is then said to be gram-positive. But if the envelope does not absorb the crystal violet stain, but absorbs the safranin counter stain instead, then the cell will appear pink-red, and the cell is said to be gram-negative. Gram-positive cell walls consist of a thick layer of peptidoglycan, which is a polymeric substance made from amino acids and sugars. In addition to its structural and barrier functions, the cell wall may also aid a pathogen by providing protection from a host organism's immune system. In addition to peptidoglycan, the gram-positive cell wall also contains lipoteichoic acid. The function of this acid for the bacterium is unknown, but the human immune system may be activated by exposure to these chemicals. Real-world example. The antibiotic penicillin targets the enzyme that catalyzes the cross-linking of peptidoglycan. If a gram-positive cell, which we just learned about, cannot cross-link its cell wall, it no longer serves as an effective barrier. The bacterium becomes susceptible to osmotic damage and lysis. Most bacteria have developed resistance mechanisms to penicillin. Although a few bacteria, including Streptococcus pyogenes, which causes strep throat and some skin infections, and Treponema pallidum, which causes syphilis, are still very sensitive to this antibiotic. Moving on, gram-negative cell walls. Gram-negative cell walls, remember, they are the color pink-red, are very thin, and they can also contain peptidoglycan, but in much smaller amounts. Okay, so the gram-positive cell wall is a thick layer of peptidoglycan, whereas the gram-negative cell wall is a very thin, smaller amount of peptidoglycan. The cell walls of these gram-negative bacteria directly abut the cell membrane. In addition to the cell wall and cell membrane, and this is important, so listen carefully, gram-negative bacteria also have outer membranes containing phospholipids and lipopolysaccharides. So gram-negative cell walls are very thin, and they have an important component, which is their outer membrane, which contains phospholipids and lipopolysaccharides. Interestingly, lipopolysaccharides are the part of the gram-negative bacteria that triggers an immune response in human beings. The inflammatory response to lipopolysaccharides is much stronger than the response to lipoteichoic acid. Just had a sip of my coffee. A key concept to keep in mind with this section. Bacteria contain a cell wall, the composition of which is different in gram-positive and gram-negative bacteria, as we just learned. The human immune system can respond to the components of the cell wall, inciting an inflammatory response. Moving on now to flagella. Flagella are long, whip-like structures that can be used for propulsion. Bacteria may have 1, 2, or many flagella depending on the species. Flagella can be used to move toward food or away from toxins or immune cells. This ability of a cell to detect chemical stimuli and move toward or away from them is called chemotaxis. The flagella are composed of a filament, a basal body, and a hook. The filament is a hollow helical structure composed of the protein flagellin. I'd like to make a small sidebar note here. Remember, the flagella that are in eukaryotes are quite different. Earlier in this chapter, in an earlier video, I read from the book and talked to you about the flagella of eukaryotes, specifically eukaryotes of motility like a sperm cell or cilia, which are found in the lungs. Cilia are not motility per se in the same way that sperms are, but they do have the flagella. Anyway, the flagella of the sperm cells are unique in that they are the 9+2 structure. The flagella of the bacteria or prokaryote are composed in a different way. So we're learning right now about the filament, the basal body, and the hook. All right, the filament is a hollow helical structure composed of the protein flagellin. The basal body is a complex structure that anchors the flagellum to the cytoplasmic membrane and is also the motor of the flagellum, rotating at rates up to 300 Hertz. The hook connects the filament and the basal body so that, as the basal body rotates, it exerts torque on the filament, which can thereby spin and propel the bacterium forward. The overall structure of flagella is similar in both gram-positive and gram-negative bacteria, but there are slight differences due to the physical structure and chemical composition of the envelope differing in gram-positive and gram-negative bacteria. Other organelles. As mentioned earlier, prokaryotes concentrate DNA in a region of the cell known as the nucleoid region, but do not contain a nuclear envelope. Prokaryotic DNA is carried on a single circular chromosome. This DNA is not coiled around histones as it is in eukaryotes. In addition, DNA acquired from external sources may also be carried on smaller circular structures known as plasmids. Plasmids carry DNA that is not necessary for survival of the prokaryote, but it may confer an advantage such as antibiotic resistance. Prokaryotics don't have mitochondria. Instead, they use the cell membrane for the electron transport chain and generation of ATP. Prokaryotes do contain a primitive cytoskeleton, but it's not nearly as complex as the one found in eukaryotes. Prokaryotes also contain some ribosomes, but the size of the ribosome in prokaryotes is much smaller than what you find in a eukaryote. That's all for now, folks. Thanks for listening.